What if a single drop of blood could tell you exactly what year your memory will start to fail?
Yeah, up to 2 decades before it even happens.
Right. Welcome to this custom-tailored deep dive. We are unpacking some incredible stuff today. Based on the stack of research you've shared with us, I mean, we're talking National Institutes of Health bulletins, network meta-analyses, and frontiers in brain. We are looking at a massive, massive leap forward in the fight against Alzheimer's disease.
We really are. We're moving away from this older diagnostic landscape that relied on, subjective memory tests or those incredibly expensive brain scans.
Or those painful lumbar punctures.
Exactly, the spinal taps. We are entering this new reality where just a simple routine blood draw can tell us exactly what's happening in the microscopic environment of the brain.
And the mission for this deep dive is to take you on that journey. We're going to start at the microscopic level, look at how these proteins behave, move to how doctors are actually measuring them in the clinic,
Yeah.
and end with this futuristic breakthrough that can predict when symptoms will start.
Because the ultimate goal here isn't just assigning a diagnosis, right? It's about human progress. It's about transforming Alzheimer's into a disease that can actually be treated or delayed or even prevented at its earliest stages.
Okay, let's unpack this. We need to start at the molecular level, which means talking about the relay hypothesis. Your sources heavily focus on two specific forms of the tau protein, and pTau231 and pTau217.
All right. So the field has known for a long time that this hyperphosphorylated tau is a major driver of the disease. But the nuance here, what these meta-analyses highlight, is that different forms of tau, the isoforms, they're doing different jobs at different stages of the disease.
Like a relay race.
Exactly like a relay.
Yeah.
And it starts with pTau231. The preclinical data shows this specific isoform elevates the earliest. It reacts to soluble amyloid beta aggregates.
Wait, soluble, meaning before the plaques even form.
Yeah, way before. We're talking about a stage so early that the amyloid hasn't even clumped together into those big macroscopic plaques you'd see on a PT scan. pTau231 is responding to the microscopic invisible precursors.
Okay, so think of it like home security. pTau231 is your hypersensitive smoke detector. It goes off at the very first whiff of danger, long before you can even see a fire.
That's a great way to put it. But, the disease obviously doesn't just stop there.
Right, so what happens next?
Well, what's fascinating here is how the biological signature shifts as the disease progresses to mild cognitive impairment, or MCI, and eventual dementia. That's when pTau217 takes the baton.
Ah, the second runner in the relay.
Exactly. pTau217 shows this steep exponential rise, and that correlates very strongly with the actual formation of neurofibrillary tangles inside the neurons.
So it tracks the real structural destruction of the brain tissue.
Yes, because it mirrors that destruction so closely, pTau217 tightly tracks with longitudinal cognitive decline. It really is the definitive marker for active neurodegeneration.
So 231 is the smoke detector, and 217 is the fire monitor that's tracking the actual blaze.
Perfect analogy.
But if I'm listening to this, I'm probably wondering, why do we need both? I mean, why not just track the fire monitor and be done with it?
Because it dictates how we actually run clinical trials. If a pharmaceutical company is testing a new preventative drug, one designed to stop Alzheimer's before any irreversible damage happens, they have to find people in that pre-amyloid stage.
So they use the smoke detector, pTau231, to find those early candidates.
Exactly. Conversely, if researchers are testing a therapy meant to, say, clear existing tangles or slow down active memory loss, they use pTau217. You use the fire monitor to track patients who are actively showing symptoms and see if the drug is working.
Wow, okay, so knowing what to look for is half the battle. But that brings us to the operational bottleneck. Because, identifying a microscopic protein in a research paper is one thing, but consistently finding it in a standard blood draw is a whole different ballgame.
It really is. It's a massive technical hurdle.
How accurately can our current tech actually measure these specific isoforms?
Well, the absolute gold standard for precision, particularly for pTau217, is called immunoprecipitation mass spectrometry, or IPMS.
Okay, IPMS, how does that work?
It's a dual process. First, they use magnetic beads coated with these highly specific antibodies to literally pull the tau out of the blood plasma.
Like a magnet fishing for tau.
Right. And then they use mass spectrometry, which vaporizes the sample into ions and measures their mass-to-charge ratio. The accuracy is incredible. It yields an area under the curve, or AUC, of 0.947 for detecting abnormal amyloid status.
Wait, let me stop you right there, because
Yeah.
you're describing, like, a sci-fi laser laboratory. Mass spec requires what? $1 million machinery? Specialized technicians?
Yes, it's highly complex and very expensive.
Right. You are not going to find a mass spectrometer in a standard primary care clinic. If we want this to be a routine test, how do we get it out of the high-end lab?
That was the big scalability issue holding the field back. The clinic-ready solution is an automated immunoassay. These are platforms that already exist in almost every hospital lab in the world.
Oh, really? Standard equipment.
Yeah. They use simple antibody lock and key mechanisms tagged with light-emitting molecules to measure the proteins. But the problem was, historically, standard immunoassays just lagged behind mass spec. They were less sensitive, produced noisier data.
So we had a highly accurate test no one could access,
Yeah.
and an accessible test that wasn't accurate enough.
Exactly, until researchers discovered a workaround called the ratio effect.
The ratio effect. Okay, explain that.
Well, rather than measuring the absolute concentration of p-tau217 just floating in the blood, they measured a mathematical ratio. Specifically, the ratio of p-tau217 to A-beta-42, or the percentage of p-tau217 compared to non-phosphorylated tau.
I am genuinely lost in the logic here. If a patient's blood sample is noisy, or, say, their kidneys clear proteins from their blood faster than mine do, how does turning it into a percentage magically make it accurate?
I know, it sounds counterintuitive, but it acts as an internal biological control mechanism. Think about the kidney clearance rate you just mentioned. If a patient's kidneys are super active and filter out 50% of the proteins in their blood, their absolute level of p-tau217 will look artificially low, right?
Yeah, which could cause a false negative.
Exactly. But those same kidneys are also filtering out the non-phosphorylated normal tau at that exact same 50% rate. Both the numerator and the denominator drop by half.
Oh, I get it. So the mathematical percentage, the ratio, remains completely stable.
Yes. The denominator acts as a built-in equalizer. It normalizes all those individual differences in metabolism. And the data completely backs this up. By looking at these ratios, automated immunoassays basically bridge the performance gap with mass spec.
That's wild. So it's like a runner's handicap in a race, balancing everything out.
Exactly. The sources note that the percentage of p-tau217 actually comes out on top, quote, by a hair, even against multiple other high-end tests. So now we have a universally accurate test that works on machines hospitals already own.
That is an incredible triumph. But it does bring up a pretty serious clinical curveball. Having an accurate test for Alzheimer's is great, but what happens when a patient walks in with early onset dementia symptoms and it's not Alzheimer's?
Right, because Alzheimer's isn't the only cause of early onset dementia. You have things like frontotemporal lobar degeneration or FTLD, which is a major factor.
And getting that differential diagnosis correct is crucial, right? Treating FTLD requires a vastly different management strategy than Alzheimer's.
Completely different. If you misdiagnose FTLD as Alzheimer's and give them an amyloid-clearing drug, it won't help, and it could cause severe side effects. This is where a specific biomarker ratio from the sources becomes the ultimate tiebreaker.
A new ratio, what is it?
It's the NFL to p-tau217 ratio, so a neurofilament light chain divided by p-tau217.
Okay, NFL neurofilament light chain. What does that do?
NFL is a structural protein inside your neurons. When a brain cell dies from any cause, be it Alzheimer's, a concussion, or FTLD NFL spills into the blood, it's a universal marker of brain cell death.
Oh, so NFL is the general siren saying damage is happening.
Exactly. And then p-tau217 provides this specific address saying, and that damage is caused by Alzheimer's.
So by putting them together, you can tell the dementias apart.
The data is amazing. While p-tau217 alone is great, the NFL p-tau217 ratio showed the absolute highest accuracy in differentiating Alzheimer's from FTLD. We're talking 90.6% sensitivity and 84.9% specificity.
What does this actually mean for a patient's experience, though, like at the clinic?
It allows clinics to use a three-range threshold approach, low, intermediate, and high risk. By using this specific ratio, clinics were able to reduce the need for painful cerebrospinal fluid testing, the spinal taps, by 54% to 75%.
That is massive. So what does this all mean? If we had tests that can replace spinal taps, rival expensive PE scans, and accurately diagnose patients, shouldn't doctors just start giving this to everyone over 50?
Well, no. And rolling this out requires some serious ethical guardrails. The AAIC 2025 clinical practice guidelines lay out very strict targets for this.
The 90-90 and 90-75 standards from the sources, right? Let's break those down.
Right. So for triage, meaning the test is just used to rule out pathology and stop unnecessary invasive testing. It needs at least 90% sensitivity and 75% specificity.
Okay, so if it's negative, you can safely say you don't have Alzheimer's and skip the spinal tap.
Exactly. But if you want to use the blood test as a totally confirmatory test, like completely substituting for a PET scan, it has to hit that 90-90 mark. 90% for both sensitivity and specificity.
That makes sense. It's a high bar to ensure they aren't causing panic with false positives. But that brings up a huge ethical dilemma from the clinician's opinion piece in the sources. What about the worried well?
The perfectly healthy people who just want to check.
Yeah. What if a cognitively normal person walks in and demands this blood test?
This raises an important question. What does biological positivity actually mean? The sources are explicit here. Biological positivity does not automatically equal a clinical dementia diagnosis.
Because of that 30% statistic, right?
Yes. Up to 30% of cognitively healthy older adults harbor these biomarkers in their brain, but they might never actually develop clinical symptoms during their natural lifespan.
Almost a third. That really highlights the danger of overdiagnosis. The clinician's piece was very firm about not abandoning clinical judgment.
You cannot abandon the clinical triad progressive decline, insidious onset, and anesthetic syndrome. That has to remain the diagnostic axis.
If we just test healthy people, we risk causing severe psychological harm, unnecessary anxiety, and totally misclassifying them. Biomarkers have to inform the judgment, not replace it.
Exactly. These blood tests are currently not recommended as standalone tools for cognitively normal people.
So you shouldn't test the worried well right now. But here's where it gets really interesting. For researchers or for people who are already showing early signs, what if this blood test could act as a crystal ball?
Moving from diagnosis to prognosis.
Yes. This breakthrough from Dr. Suzanne Schindler's NIH-funded team at Washington University. They aren't just asking, is it Alzheimer's? They're asking, when will the symptoms start?
This is an incredible Nature medicine study. They built an Alzheimer's clock model by looking at data from older adults who took multiple p-tau217 blood tests over a long period of time.
And the precision is just mind-blowing. They can predict the onset of symptoms with a median absolute error of just 3 to 4 years.
3 to 4 years. But you have to factor in the crucial context of age here. Age is the primary variable.
Oh, so.
The time from when p-tau217 first elevates to when symptoms actually appear shrinks dramatically as you get older. So if a 60-year-old shows elevated levels, they have a lot of cognitive reserve. They might not develop symptoms for 20 years.
A 2-decade biological runway.
Right. But if an 80-year-old shows those exact same elevated levels, their brain is less resilient. That window drops from 20 years down to just 11 years.
Wow. Think about how this changes the game for you, the listener. In the short term, this dramatically accelerates clinical trials to find a cure, right? You can pinpoint exactly when someone needs a preventative drug.
Yes. And in the long term, it allows patients and their doctors to build a concrete life plan. You can implement therapies or lifestyle changes exactly when they'll have the maximum impact to slow or prevent those symptoms.
It is wild to look back at the journey we just took. We started down at the molecular smoke detectors with p-tau231 and the fire monitors with p-tau217.
Then we looked at how the ratio-based automated tests level the playing field against mass spec.
We walked through the ethical guardrails, making sure we protect the worried well and differentiate dementias with the NFL ratio. And finally, we ended up at this Alzheimer's clock that can literally predict the future.
It's a completely new paradigm. We are rapidly approaching a world where Alzheimer's could be treated a lot like high cholesterol.
Just monitored by routine blood tests?
Yeah, monitored routinely and managed with preventative therapies decades before a single symptom appears. Which leaves us with a pretty profound final thought. If a simple blood draw could tell you that you were exactly 20 years away from cognitive decline, would you want to know? And how would that knowledge change the way you live your life today?
That is a heavy question to mull over. But for the first time, it's one we might actually have the tech to answer. Thank you so much for joining us on this deep dive. Keep questioning, keep learning, and keep diving deep.